Hi, everybody. It's Kim Curry here, bringing you the next steps of the MyGene project. I'm starting off here today on the BLAST site on the NCBI website. And what we're going to do today is run a protein blast of your protein sequence. And from that, we will create a phylogenetic tree. I have three examples to show you today, starting from the most likely tree example that will apply for probably the majority of you in the class. And then I've got a couple of more unique examples that may apply to some of you as well. So follow along and let's make our trees. So click on the Protein Blast site here, and it brings you to this entry page. You're going to enter the accession number for your protein sequence. Make sure it's a protein sequence. So again, if you go to your full record and you go to this section called NCBI reference sequences, we've been here before, you can get your accession number. If you have multiple variants for your gene of interest and you have therefore multiple protein products because of it, you can just choose one of the accession numbers and the site will recognize the rest on its own. So enter that accession number here. And then what I want you to do, if you're working with a human gene, as this GRM6 is, I want you to choose the model organism database here. If you are working with Drosophila or Arabidopsis or the yeast, then I want you to choose the reference proteins database here. Since this one is human, I'm going to select model organisms. And then I want you to exclude the predicted, the XP sequences from our results. We don't want to include those. You can leave all of the other parameters as the default settings, and then you select BLAST. And just like for the primer blast that you may have done already, it does take a couple of minutes for your results to show up on screen. So I've run this ahead of time to save you waiting on the video. This is what your results are going to look like, or this is an example of what your results might look like. So you'll notice here, these are all of the descriptions of the results that you can um, or that have shown up in this model organism database. What I want you to do is to have a look at the alignments tab here. This alignments tab is going to show you the alignments between the entry, the search entry, and what the result came back with. So in this case, this is our very first result. This is the same gene, GRM6. And so we can see that that's 100% identical. But as we scroll through, these sequences are going to become less and less identical, and their identity percentages are going to go down as well as their query percentages. And so you can start to look through at what some of these differences are. And if we go to one that is not very similar, you can start to see, for example, in this one here, this is a different, this looks like it's GRM8. You can start to see that there are regions of similarity, but then other regions that have changes or gaps or otherwise. So um, this is similar to how the conserved domain database identifies common regions. So once a string of amino acids is known to serve a function or a purpose, structural purpose, for example, and it shows up in other proteins, then it can be predicted that it serves the same function or structural purpose. So let's scroll back up again and let's have a look at the descriptions. You'll notice that automatically all of the options, all of the descriptions are all selected. I want you to deselect all of the options and I want you to look through your list to see. So we start with the human. What I want you to do is select the organism, same organism, as it shows up for the third time. So what I mean by that is we have humans here. Then we have other organisms, doesn't matter what they are. Then we have humans again. So that's showing up a second time. And then we have a different organism. There may be many in here for yours, doesn't matter. And then we come back to humans again. So what I want you to do is to select up to 3. So as it cycles back to the same organism a third time. So humans, different organism, humans, different organism, oops, different organism, and humans again. Okay, so that's how many you're going to select. It's going to be a little bit different, obviously, for each gene. And I have upcoming examples that don't exactly follow this rule. So if yours doesn't quite look like this, keep watching. Then you're going to select the distance tree of results, and it is going to produce for you your phylogenetic tree. So what we're looking at here for GRM6, so highlighted is going to be the sequence that you put into the BLAST tool. And this tree, if we look carefully, we can see there's human and mouse GRM6. So this is showing a speciation event here. And if we look here, we have what looks like GRM 6A and 6B. And so this here is a duplication event in the Danio rario, which is a fish. So here, this is going to be a speciation node. And if we look up on this region of the tree, we see what looks like GRM8, so a slightly different receptor. And these two mice isoforms, these are just variants from the same gene. So these are not duplication events. And then we have humans. We have 3 variations of this GRM8. Again, these are isoforms, so they're not duplication events. So that means that there is a speciation event that occurred here, and there is a duplication event here when it's split off into GRM8 and GRM6. And again, there's one more duplication event here where it was GRM4. So don't worry too much yet about these duplication and speciation events. Maybe have a look, see if you're able to identify them. But we're going to save the following week, week 7, to really diving into the details of each of your trees. This week, I really just want you to focus on making the tree and having it available. So how are we going to save this? You're going to go to Tools, Download PDF file. You select Create PDF file. It creates this link. And here you have a PDF version of your tree. So download and save a copy of this. This will also be very useful for you to use in your presentation at the end of term. So that's an example using the human GRM6. Just to give you a quick summary, when you do a BLAST-P with a human, you're going to use the model organism database. You're going to exclude any predicted sequences. And when you have your BLAST results, you're going to start with your organism. It's going to be the first hit usually, always. And then you'll leave to other organisms, come back to the same one, leave again, and come back. So that's how many you're going to choose to include for your tree of results. Now, not everybody will see this nice, lovely list, and this rule may not apply. So let me show you a couple more examples using different sequences. So in this example, I am going to use this FLC gene, which comes from Arabidopsis. So this is not a human sequence, which means that I am going to use the reference proteins database here, this one. I'm still going to exclude the predicted sequences. And when I run the BLAST program, I hit BLAST. As I mentioned, I've done it ahead of time to save time. We'll see that this list looks a little bit different than the GRM6 one just did. So these are the same results here, but the same rule does apply. So what you're going to do, you're starting with Arabidopsis. You have a different organism. You have Arabidopsis again. different organism, and Arabidopsis. So these are all of the ones that get selected to be included in your distanced tree of results. So here is the tree for the FLC Arabidopsis gene. So save this PDF and we can examine this in the lab the following week. This one is quite interesting. There appear to be lots of different duplication events in this one here. So this will be fun to explore in the following week. Now let's have a look again at even another example. This one will be the leafy gene. So this is a leafy gene from Arabidopsis. And when we run the BLAST protein search, these are the results that show up here. And so for some of you, you may find that there is your organism here on the list, but it's only at the top of the list and it doesn't show up again. In this example, there's only 16 sequences in total, but none of them loop back to Arabidopsis. So in this case, I want you to choose the top 10 results here. So these most likely are from 10 different organisms or very close to. And then we'll do the distanced tree of results. And here we have the resulting phylogenetic tree. And so this one also is quite interesting. These sequences here are both variants from the same gene, so these are not duplicated. And then you can see that there are many different organisms also shown in this tree as well. And so this gene actually doesn't appear to have a duplication event, which in itself is actually quite unique, but there are lots of demonstrated speciation events in this tree. Again, we'll dive into these details and the specifics of each tree in week 7. We'll just focus on creating these for this week. So certainly everyone's results, as to be expected, are going to be unique. So we will help you in the lab to choose if your gene doesn't apply, if these rules don't apply for your gene, the 3 different options I've gone through with you here, then certainly we'll go through it with you during the lab and help you in selecting which results from the protein blast should be included for your distance tree of results. So just as a reminder, final review, when you're using the BLAST-P, you want to use model organism database. If you have a human gene, you want to use reference proteins if you're using any of the other 3 organism options for this term. So that's Drosophila, Arabidopsis, and P. Saccharomyces. All right, thanks everybody. Have fun making your trees, and I cannot wait to see them.